Starch, you know, dextrine is a shorter form of starch, partially digested. Glycogen, you know, anybody, any idea about this? Huh? GFR. For GFR, what is it made up of? It's made up of fructose. It's a polymer of D-fructose. Good. Dextran, maltose, galactose. It's a dye. Agar, again, it's a polymer of galactose. It is found in seaweeds, agar. Agar, did you ever have agar? Yes, in which form? Very good. The jelly kind, jelly-like of things, custards and those things, so they add. Good, very good. Cellulose. Did you have ever had cellulose? Vegetables full of cellulose, right? Very good. Difference is glycogen and starch and dextrine, they contain D-glucose, alpha D-glucose, and cellulose contain beta D-glucose. We'll come to that beta D everything, don't worry. Since you are responding very fast, so I can go a little faster. So heteropolysaccharide examples are like this. The major example of heteropolysaccharide falls in a separate group called mucopolysaccharide, which is specially designed for our biological system. Apart from that, we have many other heteropolysaccharides. So for example, lectins, lignins, right? This is a group of compounds, lectins. Now the first long question we have finished from this chapter, that is classify carbohydrate. Okay, the detailed example will come as we will progress, but more or less the first question we have finished, that is classification of carbohydrate. The second long question comes from this chapter is isomerism of carbohydrate. Correct? Now this is the isomerism, need not to write, don't write, just go through. We have studied in class 2, correct? And you should know about all the terms here. Fine. So in this chapter, in particular in the chemistry part of the biomolecules, many times you will come across these problems, these terminologies. So then you should be able to correlate what I am talking about. Structural isomerism is something different, stereoisomer is something different, diastereomer, racemers, geometric isomerism. Geometric isomerism, what is that? The position of the functional groups or additional groups on the same side of the bulkier groups on the same side of the double bond, then we call it cis. Otherwise, we call it trans. Very good. Optical isomerism, any idea? R, S configuration, R form, S form, correct? D and L isomerism, right? Fine. Different spatial arrangement of the functional group. Very good. This one, chain isomerism, N-butanol, isopropanol, N-butanol, isopropanol. Why I am talking about this? This is not chemistry class. Do we use anything to wipe out before taking injection? What is that? Alcohol? What alcohol? Isopropanol. Correct? Position isomer. Position isomer. Any example? The double bond can change. The functional groups can change. Example, one butan rural gel, আমি বায়োমলিকুলস থেকে বলছিলাম, যেমন বিটা অ্যালানিন, এটা অ্যামিনো অ্যাসিড, তাই না? ফাংশনাল গ্রুপটা বিটা অ্যামাইনো গ্রুপটা বিটা কার্বন থাকে, আলফা কার্বন থাকে না, তাই না? কিন্তু অ্যামিনো অ্যাসিড কিন্তু আলফা অ্যালানিন, এটা হচ্ছে বিটা অ্যালানিন। Now regarding isomerism, these 5 forms we must remember. At least you can write as many as possible, but these 5 should be sufficient to get you good marks. If you can explain these terms under the heading of isomerism of carbohydrate. Clear? First one, D and L isobound. Second one, small d or plus and small l or negative. Third one, anomerism. Fourth one, epimerism. And fifth one, pyranose and furanose forms. Right? Should we start? Right? Hope you understand most of them, but still, if I ask you to describe the shape of this object in my hand, what is the shape? Don't lie. From your side, you can't see it is cylindrical. Yes. Now, rectangular. That's what you say. We are in the 3D world. We cannot express things in 3D. We express one dimension less in 2D. Had our ancestors been migrated to a different planet or system, other than solar system, where it was a 4D world, then we would have started drawing 3D diagrams in our copies. But copies are 2-dimensional. So this is our experience from the birth. We have come to know that when it looks like this, it is cylindrical. The light and shade makes us perceive that this is cylindrical. If our previous information regarding this tells us it is cylindrical. Correct? The person who is blind, if he is asked to describe elephant, which part he will describe? The part he will touch. If he touch the tail, you will say elephant is hairy. Correct? Like a caterpillar. Correct? If he touches the trunk, then it is cylindrical, like a snake. Simply. So the problem arises when we describe a molecule and try to draw on paper. So Fisher said, when you draw on paper, we keep the last but one carbon atom, OH group, either on right hand side or on left hand side. If it is in right hand side, it is D form. If it is on left hand side, it is L form. So this is what you have to describe. The penultimate carbon, the last but one carbon containing OH group on right hand side is called capital D, D form. It has nothing to do with small d. It's completely different. Okay? Almost all compounds can exist in D, capital D and capital L form. It is just orientation by which we represent. They never exist in this way. The molecules don't bother about how we represent them. They are on their own. We, poor fellows, we don't know how to express. So we are trying different ways. Like a God, Almighty. Someone calls his Krishna, someone calls his Allah, someone calls his Jesus. Because it is so big, we are blind. We are perceiving like an elephant. The portion, whatever portion we touch, we come in contact with, we describe the whole thing will be like that. But it is not like that. The God never bothered to come and correct us that if we are making any mistake. He never bothers. If an ant climb up to an elephant body, will the elephant come to know? Never. Simply, we are like smaller than ants and God is bigger than elephant. Fine. Simply, so molecules are like that. They're in that level. They don't bother about what we are thinking. They exist in their way. We try to find out which form they are in. So there we describe, am I able to convey the message? It is not the molecule that who will bind by the laws made by us. We will make the laws seeing the molecules that might follow these laws. Correct? So, D-glyceraldehyde means OH will be on right hand side in the pentyl carbon, L-glyceraldehyde OH will be on the left hand side. Fine. Have you ever seen, is there anyone who has seen this series in any book in class 2? Which book? Which sir? S. Chand? Bell and Bell chemistry book? Huh? Bell and Bell? Very good. Is there anyone who has ever turned the pages of Finner? IL Finner? So actually this diagram was taken from Finner. Bell and Bell took this diagram from the Finner. Finner was first one to give this diagram, which shows the series of sugars of D series, aldose sugar, starting with D-glyceraldehyde. Now in this diagram, it is very clumsy. There are so many names, and except I think first one or two benches, nobody can read it also. Correct? But we can remember the name, we can draw this whole series on our own. Is there anyone who knows the technique? If not, then I am going to describe. If yes, then he or she can describe. Huh? Okay, fine. Let me describe then. How to remember this series and why to remember this series? Because many times a question comes regarding the structure. So if you remember the structure, it is very easy to draw. If you know how to draw a structure, you can draw any structure. But if you don't know, then you have to remember memorize each and every structure. Name by. Huh? The first one is D-glyceraldehyde, which is 3 carbon containing trial sugar, D sugar, L sugar. D-glyceraldehyde, L-glyceraldehyde we are not considering. We are having only D sugar. Now each time in one step vertically, when we are going one step down, we are increasing 1 carbon by number, and the newly added carbon is adding at C1 position. These two we noticed. Each time we are going one step below, from 3 to 4, the new carbon is being added at C1 of the newly formed compound. And by convention, when we write on the compound on left hand side, we put, see, this one, the C2, which was then before C1, now it has become C2. So it is a new chiral center. The new chiral center, OH might be located either left hand side or right hand side. This is convention made by all the chemists that when we draw the structure like that, the left hand side structure should have OH on the newly formed chiral center on right hand side. নতুন chiral center এ OH টা ডান দিকে থাকবে আর বাঁদিকে ডান দিকেটার OH টা বাঁদিকে থাকবে। তাহলে বাঁদিকের কম্পাউন্ডের OH টা ডান দিকে আছে, ডান দিকের কম্পাউন্ডের OH টা বাঁদিকে আছে। Rest all will remain same. Why? Because it's a D series. The last carbon, penultimate carbon will not change. Last 2 carbon will not change their configuration. Only other carbons will change. So now it is increased by 1. So now this is tetrose, 4 carbon compound containing compounds. Next level is pentose, next level is hexose. Now how to remember the name? The first one is D-glyceraldehyde, second one is erythrose and threose, ET. Just write these mnemonics, ET, ET, extraterrestrial, so that ET, erythrose and threose. Left hand side erythrose, right hand side threose. Next level, pentose sugar, if you are coming from erythrose, 2 pentoses will be there. From threose, 2 pentoses will be formed. Name them. Ring all xylophone loudly. Pentose sugar, ribose, arabinose, xylose, lysose, LYXOSE. Right? Lysose. For pentose sugar, for hexose sugar, nothing is made by me. All are given in chemistry book. Don't think I am very knowledgeable person. I just gather knowledge before you. That's all. All alchemists gladly make gum in Galen tanks. Aloes, altrose, gladly, glucose. Then make mannose, gum, glucose, in, iodose, Galen, galactose, tanks, talose. Remember the name? Structure you can draw anytime. You should know how you draw the structure. You can draw structure anytime. You can draw innumerable compounds. The compounds may not exist in nature, but you can draw their structure. Fine? Any question? Correct? Fine? Similarly, for ketose sugar, do we have any series? Yes, we do. For D-ketose, we start with dihydroxyacetone. The first member, dihydroxyacetone, triose, keto-triose. Next step, it forms only one form. Other form is not available. Tell me why. In this case, the newly added carbon is at C2, correct? And the next carbon should describe its isoform, right or left. The next one cannot be right left. Why? Because it is D series. So we don't have any corresponding sugar for thiose in keto series. We have only erythrulose. Fine. So that's why naturally the next step it will be less again. Instead of 8, we will have only 4, we will have only 2 ribulose and xylulose. This was Cysose, P remains silent. Cysose, fructose, sorbose and tagatose. So here we know where is the fructose, where is glucose in the series. So if we call what we name one aldopentose, arabinose, name one ketopentose, xylulose, name one ketohexose, fructose, sorbose, cysose, tagatose, correct? If they ask you to draw the structure also, you can draw. It may take some time, but you can draw. Correct? You need not to memorize the structure anymore. If you remember the method. Correct? Now, this is assertion, rule. In the present system on earth, in our body, we find most of the sugars in D form and amino acids in L form. The opposite is possible? Question mark. Yes, possible. Now also it is there. But it is made with an goal to make it resistant against the enzymes produced by us, because our enzymes, they can digest D sugars containing substances, L amino acid containing substances. We cannot digest compounds formed by D amino acid and L sugars. So where possibly could you find these compounds? The compounds containing D amino acids and L sugars. The cell wall of bacteria. They want them to be protected from the enzymes of our body. Our enzymes can digest only compounds formed by L amino acid and D sugars. So they make their outer part of the cell wall with this, so that we cannot digest the outer part of the cell. Right? But inside they utilize the same nutrients what we use. So their metabolism runs on with this. But outside protection is like that. So that's why many bacteria we cannot digest. But there are so many bacteria we digest. How? We have a specialized enzyme called lysozyme. We have a specialized enzyme called lysozyme. Lysozyme can cleave that particular cell membrane, cell wall. Right? Even we have protection. Right? They are not invincible. Fine. So there are some important structures. Anyway, we'll come to that structure later. We'll see D and L refers to. Let's go through it. Already I have described. 2N rule and all these things are not required to remember now because you are studying medical biochemistry. Now, anomers. Anomers. Anybody is having any idea about acetal and hemiacetal, ketal and hemiketal? When aldehyde and alcohol combine, they form acetal. When keto and alcohol combine, they form ketal. When these two molecules, functional group, are part of the same molecule, it is called hemiacetal or hemiketal respectively. কি বললাম একই মলিকুলের মধ্যে অ্যালডিহাইড এবং কিটোন গ্রুপ অ্যালকোহল গ্রুপ থাকলে তারা যদি কম্বাইন করে তাকে বলা হয় অ্যাসিটাল। একই মলিকুলের মধ্যে কিটো গ্রুপ এবং অ্যালকোহল গ্রুপ থাকলে তারা যদি কম্বাইন করে তাকে বলা হয় কিটাল। এবং যদি এই দুটো গ্রুপ একই মলিকুলের মধ্যে থাকে, সেম মলিকুলের পার্ট হয়, তাহলে তাকে আমরা বলবো হেমিঅ্যাসিটাল অর হেমিকিটাল রেসপেক্টিভলি। Now this is a molecule, example of molecule which has aldehyde group and many alcohol group. So if the C1 aldehyde group combines with C5 alcohol group, then it forms this kind of structure. Now the problem is this combination of these two groups has generated a new chiral center at C1. C1 is now no more a non-chiral carbon. Now it can have OH group either on right hand side or left hand side. So if they have it on right hand side, we call it alpha. If they have it, right hand side, we call it alpha. If they have it on left hand side, we call it beta. While drawing in a pyranose form, the right hand side OH goes below the plane. Right hand side OH goes below the plane alpha, and left hand side goes above the plane beta. Correct? So anomers can be of only two types, alpha and beta, and that is formed when they form a ring structure within the same molecule. When carbohydrates form a ring structure within the same molecule, a new, this is the thing, this is the wording you should write simply. A new chiral center is generated when a carbohydrate forms a ring structure within the same molecule, and these chiral center may contain OH group on right hand side or left hand side. If the OH group reside on left hand side, we call it beta. If the OH group reside on right hand side, we call it alpha. While drawing, it goes below the plane and above the plane. In case of alpha, below the plane. In case of beta, above the plane. Clear? So anomers are of two types. So this is for glucose and this is for fructose. In this case, it has C2 and C5 has formed the linkage, right? So one oxygen is there and this furanose ring structure is formed. The 6-member ring is called pyranose form, while 5-member ring is called furanose form because they resemble the heterocyclic compound pyran and furan respectively. This is something which we do not want to discuss now, not required. We go to next form, epimers. This alpha and beta isoform of glucose is very, very important. Very important. For our everyday daily life, it is required. We digest, we metabolize alpha form, not beta form. And in laboratory, we measure beta form, not alpha form. It's simpler. It requires less money. So when you are doing something measurement, estimation, you should see about the profit. That's all. You will come across many things like this glucose. We'll measure alpha, we measure beta form actually in laboratory. And then vitamin D, actually we write it's D3, but we measure not 125 but 2425 D3. Because 125 measurement of 125 D3 is more costlier than measuring 2425 D3. When you write it is D3, but the position is changed, functional isomerism. Right? Simple things. Fine. So epimers are those isomers who differ in their structure at any carbon atom. Any carbon atom, not C1, C2 specified. Any carbon atom. D and L? Yes, enantiomers. D and L. So according to that, anomers are also epimers, but at C1 in case of aldoses and at C2 in case of ketoses. Fine. So examples, epimers, two beautiful examples are there. Glucose and galactose are epimers at C4, and glucose and mannose are epimers at C2. You want to see the structure? Glucose and galactose. Check out at C4. See, all the structures are same configuration except C4. Check out glucose and mannose at C2. See, try to remember, see and remember. Right? Good. Now, what type of glucose is alpha or beta? Alpha. Good. Very good. And this one, mannose? Alpha. Very good. Brilliant. Ring structures. Easier than ever. Easiest. One who understands all these things, everyone, for them, ring structure is very easy. Pyranose and pyranose. Fine. Then why did we include this one as important? There are many other forms which have excluded, but we have included this one. Why? Because of some biological property. That is, glucose exists in our body in pyranose form. Fructose exists in our body in mostly pyranose form. That is why. Clear? Now we come to other oligosaccharides. Raffinose. It's a trisaccharide. Just know the name and know few function. What is function? No oligosaccharide has any function to provide energy. It is only dietary fiber, components of dietary fiber. Raffinose. Where do you find? This is the structure of raffinose. What is structure? Very easy. Can you tell me what is this structure? Sucrose. This is glucose and this is fructose. So glucose and fructose combine to form sucrose. To that, at 6th position, if you add one galactose, it becomes raffinose. What is raffinose? It is found in vegetables. See, D-galactose and sucrose. Mostly found in, not found in the digestive tract. Right? Now, next is stachiols, tetrasaccharide. We find the same again in vegetables. Structure. Add one more galactose to that. Very simple. Add one more galactose to C6 of the existing raffinose. It becomes stachiols. Fine. Next is pentasaccharide, means Roman 5. Roman 5 means with what? B. starts with V, verbacose. So verbacose is a pentasaccharide. It is also found in vegetables similar to raffinose and satiose. Cannot be digested by human, but it has got an extra property. It can be used as an artificial sweetener. Artificial sweeteners are those compounds which will give sweet taste but will not give any energy. Structure, just add one more galactose in the same way to statiose, it will become verbacose. So now you know the examples and structure of at least one trisaccharide, tetrasaccharide, pentasaccharide. Beyond that, none of us know, no one will ask. Correct? Once you know the limitation of the teacher, you can make your limitation for studying also. You know? Fine. These are sugar alcohols. Ribose, when oxidized to form, oxidized or reduced, sorry. Reduced, it is formed ribitol. Now, this is gluconic acid and this is glucuronic acid. What is the difference? Very simple difference. See, what is the difference? See and tell. Huh? At gluconic acid, C1 is oxidized. C1. Correct? Glucuronic acid, C6 is oxidized. Means first one is aldehyde to glucose, one step oxidation. Second one, alcohol to glucose, two step oxidation. Now, is it possible that you are going for two step oxidation of C6 and C1 will remain unoxidized? Is it possible? Is it possible? Can you reach number 0 from number 3 without touching number 2? No, not possible. So how do you achieve it? If you add bromine water, mild oxidizing agent, then glucose will be converted to gluconic acid. If you add concentrated nitric acid, strong oxidizing agent, it will form saccaric acid, glucaric acid. Both C1 and C6 will be oxidized. Then how do you convert only glucose to glucuronic acid? It is not possible by any chemical method unless you bind C1. You have to hold C1 so that it cannot get oxidized. How? Only done by metabolism. We have a separate pathway for conversion of glucose to glucuronic acid, mannose to mannuronic acid, galactose to galacturonic acid in our system, our metabolic system. Okay? Uronic acid pathway. And these uronic acids are very, very important for making so many compounds. Sometimes the sugars are also derivated to amino sugars. So which position they are mostly added? See, either they are added, see, 2 or 4 position by simply make it glucosamine, or they are added, sometimes it is also added with one acetyl group. So it becomes N-acetyl glucosamine. Mostly at C2 position. Mostly at C2 position. Remember, glucosamine and N-acetyl glucosamine. Sialic acid. You might have heard this term, sialic acid. This is the structure of sialic acid. See, what I told you, 2 and 4, mostly amminated. See here the example of 4 position amminated and 1 acetyl group is added. So this is sialic acid, N-acetyl neuraminate. This R group is this. R group is this. What is this group? One glycerol. One molecule of glycerol. So you add one molecule of glycerol at C6, you add one molecule of acetylated amine group at C4, it becomes N-acetyl neuraminate. It becomes a 9 carbon containing compound. Sialic acid. 6 plus 3, 9 carbon. Correct? So, example of one derived sugar, example of one acetylated sugar, example of one nonose sugar. One example. Sialic acid. Okay? Clear? Okay, glycosidic bond will occur nahi. Now the structures of detailed structure of disaccharides. Sometimes the question asks. Sometimes disaccharides, we will restrict our knowledge, our study within very few compounds. So we'll start with to make a table as we go on. Just write down, fill up the table of disaccharides. Okay, what are the columns? Columns will be components. Name of the disaccharides is separate. Then first column will be components. Next linkage. Next digestive enzyme. And next one you keep for some other points. 4 columns you make. Okay. Ready? When you just tell me when you are ready. Okay. Fine. Start. Fine. So first one is maltose. You have heard of maltose. Maltose. What is the source of maltose? Any sugar, any naturally occurring sugar other than cane sugar. Other than cane sugar. Okay. Molasses and etc. etc. Gur. So there, or partially digested starch, Fine. Components glucose plus glucose. Linkage alpha 1,4 glycosidic linkage. Enzyme maltase secreted by brush border cells of small intestine. Mucosal cells of small intestine. Hello? Fine. Uses, you write anything, no one asks. Next, isomaltose, made up of glucose and glucose, linkage alpha 1,6, digesting enzyme, isomaltase, so location, secretion source is same, brush border cells of small intestine, and function is not much. These are found in the branch points of undigested product of starch. 1,6 linkage and glycogen. Next is sucrose, glucose plus fructose, linkage beta 1, 2. Actually, we should write beta 2 and 1 because fructose remain in beta form and glucose remain in alpha form. But since we prefer a sequence 1 and 2, so we write, mostly books you will find writing beta 1, 2. Actually, beta should be beta 2, 1. When you are telling 1, 4, 1, 2, how you write? Yes, there are 2 ways of writing. One is, another is okay, clear? Any of this method you can follow. Okay, sucrose is done. Then enzyme is sucrase. Then lactose. Glucose plus galactose. Beta 1,4. This is not alpha 1,4. This is beta 1,4. Beta D galactopyranoside, alpha D glucopyranoside. The name. This is called every name also. Beta D galactopyranoside, alpha D glucopyranoside. The complete name of the compound. Anyway, so lactose is made up of glucose and galactose joined by beta 1,4 linkage. So here we require beta lactase. Lactase enzyme is different from alpha amylase. Beta lactase. Correct? Next. Now here some clinical correlation is present. Lactose intolerance, a disease called lactose intolerance. Just write lactose intolerance occurs in absence of lactase enzyme. We'll come to that in detail. Lactose intolerance occurs in absence of lactase enzyme. Hey, bullish. Oh, acha. The structure of starch. There are two parts: amylose and amylopectin. I think this part I will just go through very rapidly because all of you know. Amylose and amylopectin, just difference wise, if you remember, amylose comprises a structure of helical structure. Amylopectin is a branched structure. Amylose is water soluble. Amylopectin is water insoluble. Amylose contains alpha-1,4 glycosidic linkages. Amylopectin contains alpha-1,4 and alpha-1,6 glycosidic linkages at the branch points. Amylose give deep blue color with iodine solution. It's a type of physical reaction where the iodine molecules gets encaged within the spiral structure of the amylose. When heated, its structure disrupts, the iodine gets out of the structure, the blue color disappears. When cooled, the iodine again enters the structure of the helical structure of the amylose and color reappears. Right? But similar action doesn't take place with amylopectin. Here, which part of the rice we eat, amylose or amylopectin? We throw away amylose and we eat only amylopectin in the form of rice bran. Correct? Amylose is 20%, amylopectin is 80%. So which is insoluble after boiling rice that we take, that is amylopectin, and we throw away. Suppose we purchase the rice of amount of 100 rupees, we throw away 20 rupees after boiling and eat 20 rupees, 80 rupees. Right? Anyway, so aging process, when you age the rice, you must have seen the ads, basmati rice aged 2 years, 1 year. Why? The aging process removes the amylose. Amylose is easy to digest, ready to provide energy, but ready to provide energy is not good for our health. We'll become fat. So eating rice will be less. We cannot eat more rice. If we need to eat more rice, then we need to reduce the amylose. But moreover, the presence of amylose makes the rice sticky, which is not liked by most of the well-to-do rice eaters. Correct? So they prefer aged rice than the newly harvested rice because the amylose part would be less by the bacteria. So we take the aged rice. Basmati is more costlier than freshly grown basmati. Correct? But so it is a difference, that's all. Okay. Then any question about starch structure? No. Fine. Suppose we add rasgulla. You heard rasgulla? So in rasgulla, there is a test, easily done test to identify any adulteration in rasgulla. So usually in rasgulla it should be made up of chana from milk, but to increase profit or reduce cost price, they add flour, maida into it. So when they form rasgulla, maida remains in it. How to test the rasgulla is free from maida? Just carry one small bottle of iodine solution and add 1 drop of iodine solution on the rasgulla purchased from the shop. If it turns blue, that means it contains maida, starch. If it doesn't turn blue, good. Right? It's a test. Simple test. We stop here? No. দেখো আমি বলি তোমাদের নতুন কারিকুলামের টাইম টেবিল অনুযায়ী 9 to 9:50 I should take that class. To stop here or should we continue for 10 minutes? না সবার ওরকম বললেও হবে না, সবাইকে বলো, তারপর কাছে ফিরে মার খাবি, সবার হাতে ভালো হবে না. If everyone agrees, I will take 5 more minutes. If every one of you agree. Are jore bol. Thank you for giving me 5 more minutes. Fine. So we'll go to glycogen structure. Let's see what is there. Glycogen structure is unique. It is somewhat like amylopectin, but it is more branched than amylopectin. It is somewhat like amylopectin, but more branched than amylopectin. Ideally, when you view glycogen molecule under microscope after staining, we see like this. Highly branched. Okay, so glycogen is a storage form of carbohydrate, polysaccharide. In the animals, we are animals. We do have glycogen in mostly in few tissues it is stored. First one is liver, second one is muscle, third one is kidney, fourth one is brain, fifth one is testes. But other except first two, rest of the organs we do not mention mostly because of their low amount of glycogen storage. Mostly two tissues, liver and muscle. Now liver stores around 10% maximally of its weight in the form of glycogen. Muscle stores around 1 to 2% maximum of its weight. So which tissue contains more glycogen? Liver. Sure? One is muscle. What is the muscle mass of an average man? Average man, muscle mass? 35 kilo. Half of the, 70 kilo, half. So 30 kilo. If you ask physio sir, he will reduce further. Okay. Okay, 25 kilo. That's all. Below that I can't go. 25 kilo. Now you count for 25 kilos of muscle, and then you count for 1.2 kilo of liver. Count and tell me what is the value. Go ahead. Mobile. কত হচ্ছে বলো কোনটা বেশি আছে 1.2 কিলোগ্রাম লিভার হ্যাঁ স্ট্যান্ডার্ড লিভার আর তার মধ্যে 10% কত হবে 120 গ্রাম সহজ তারপর ওখানে 25 এর মধ্যে 1% হলে 25,000 1% কত 250 গ্রাম এটা দোকানে একটা অশিক্ষিত সবজি আলা করে দেবে ওর অবস্থা খুব খারাপ তোদের বাড়ি কোথায় বর্ধমানের ছেলে তুই এটা এই হিসাব করতে পারি না হ্যাঁ বস তাহলে muscle stores more amount of glycogen but muscle glycogen is called selfish glycogen. Function of these two glycogen are different in our body. The liver glycogen provides energy between two meals. MEALS দুবার খাওয়ার মাঝখানে সকালে খেলাম 8টার সময় বিকেলে খেলাম 4টার সময় কতক্ষণ গ্যাপ হলো 8 ঘন্টা তাই তো up to 4 to 6 hours from the meal the liver glycogen can provide energy beyond that it stops but next 2 hours I am still working something is maintaining our body energy and that is by gluconeogenesis but here for next between two meals we should have taken food by 1 o'clock right so between two meals usually it maintains the blood glucose level which one the liver glycogen but muscle glycogen fails to do so because of the absence of one particular enzyme glucose 6 phosphatase that is why muscle glycogen is called selfish glycogen because you may die due to hypoglycemia muscle will not donate a single molecule of it will donate a single molecule but not enough molecule of glucose muscle might donate very small amount how much maximally about 1 molecule of glucose for every 8 molecules of glucose present in a glycogen. There is a calculation. But that is too little and insufficient. It is almost negligible. So muscle glycogen is that's why you call selfish glycogen. Clear? Muscle glycogen does not require a specialized protein called glycogenin for its synthesis. Liver glycogen requires glycogenin for its P-synthesis. Glycogenin. Okay. What is the reaction of glycogen with iodine solution? It turns red. Starch turns deep blue. With what type of blue? Prussian blue. Does anyone have Prussian blue? Her dress color is Prussian blue. You can see that kind of blue. And red. Do you have red? Yes, the color. That kind of red color you'll find with glycogen. Any question so far? That's all for today.
You are going to be a doctor, you are going to be a MBBS doctor, etc. when I study. But there is a specific adjective just before your doctor, that is the Indian medical graduates. There are some facets, some attributes is been imparted our National Medical Commission to you. Just having a patient, examining and giving treatment, it is not the motto of your to be a doctor. There are so many facets. You should be a good clinician, you should be a good communicator, you should be a lifelong learner, and you should be a social worker also. But whatever it may be, the basic thing is same. Actually, we learn. We are having a goal, that is going to be a doctor. So why we learn? Mainly anything. আমরা যে কোনো জায়গায় কি motto থাকে তিনটে? What is the motto? Having learning and acquiring knowledge. Suppose this anatomy subject. What is your motto? Can you tell me? Why you are reading anatomy? Anything, tell me. Huh? हाँ बोलो ना, दादी बोलो. No. Yes, very good, both of you. To know about the subject. To know the subject so that you can apply when you treat a patient. Anything else? To know an application. That's good. But the hidden thing is that you have to pass the examination. Jai koro, no, you know everything, nothing, but you cannot tell, you cannot write, theory paper. It is a zero. So, though you will not tell in front of others that it is my motto, you will tell what it told, that is to acquire knowledge, to apply it. But your hidden is that to pass the examination, ultimately. Otherwise, it is a big zero. And now, from this, already you know, they say there is no supplementary examination. You have to wait for 1 year, long year, if you get plucked. Specific to your knowledge. You must be concentrate your knowledge so that you can get through the examination. Right? So, the most important thing, how to read, what to read, and how to apply it in examination. So, and for that reason, time is very important. You start reading from day to day. Otherwise, you cannot cope up with. You have to take some unfair means in examination. But you can easily, and I am telling you, I am again telling you that anatomy is a very easy subject. Anatomy is a very easy subject. Just you follow my dictum and try to write in English. Those, suppose we are not familiar with English, in anatomy writing, not very much English knowledge is needed. Just it is known as, it is divided by, it goes through, it pass under, it rests on. This simple term is very important, right? And you must be specific from this day. Thank you. From this day, you choose the book and try to write try to read this book singly from today to last one. You choose a book and read it. Not so many books consult. Suppose if, for example, in general anatomy, I will tell you that what other books you have to read is something the teacher will, the faculty will tell you, right? So, in this way you start writing. Now, already 10 minutes passed, I am not now going to details. So, first is the introductory class, anatomy, some aspect of history of anatomy. Again, I am telling you, not very important in your examination, but just to acquire knowledge, you have to write. You know, there are so many, anatomy dates back for everything. Even the ancient, the nomads, nomadic people, the hunter, so many thousand years back, know the anatomy of the animal, so that they hit the stone or some the arrow on the vital point of an animal, so that they have got some idea. They hit either the head or put the stone or the arrow or something like that, their ancient weapons, to the heart region of the animal. So they have got idea. The missionaries, Chinese, the ancient people have got idea, but these are not refined. Ultimately, there are so many anatomy knowledge in our India also, but this is not written properly, so that we cannot expose it, we cannot tell it. But and ultimately, so the Western culture gets upper hand. So we know about the Western culture. So I'm not going to the other details. You know the Hippocrates, the great scientist, Greek physician who studied anatomy and physiology, and he is regarded as the father of anatomy. So Hippocrates of Kos, in the Greek region, he has took birth in the Kos of the group. So he is called the Hippocrates. Actually, is the Hippocrates II, Hippocrates of Kos, and much of his work today. And you have to you have to take the oath, Hippocratic oath, after completion of your MBBS study. And another, Hippocrates is known as not the father of anatomy, it is written here, is the father of the medicine. But Herophilus is called as the father of anatomy. He is one of the first to be detected human body, Herophilus. Just note down the name, not very in detail. And Galen, who also dissected the body, not very much important. I am a man of working together how to get past. So that what I told you to write down, you will write down. Just having a knowledge, Galen of Pergam and Andre Vesalius have dissected the vessels of our body, have a good knowledge about the heart. These are the, and you know, who is it? Okay, thank you very much. It's an exceptionally, sometimes I think whether he's a human being or not. Excellent, everything he has got is his own bag. So he is everything. So Leonardo da Vinci, this drawing is so beautiful drawing. Look, it is written and the drawings of the Leonardo da Vinci, and previously how the anatomy classes were taken in the English era, in the Middle Age. And this is the dissection hall of the Middle Age in the British, Britain, how the dissection and the classes were taken. Not so many people are there. Very beautiful dissection. Still in Medical College, Kolkata, this type of lecture halls are present in MCU, because I have retired from the Medical College, Kolkata, so I know. And it is the first book of Henry Gray, the first edition book of Gray's Anatomy in 1898 possibly. It is, for that 1859. Sorry. So it is the first edition of the book of Gray's Anatomy. And it is the recent edition of Gray's Anatomy, 42nd. And he is the great. We have to give a namaskar, pranam to him, because he is the Madhusudan Gupta. He is the Madhusudan Gupta, pioneer of the Indian dissection. He, in the Medical College, Kolkata, dissected the body, first cadaver. It is not so easy in that period. There are so many superstitions, so many obstruction by an Hindu, by an Indian to dissect the body. He overcome all the dissections, all the hurdles, and first time in India dissected the body in Medical College, Kolkata. He is the Madhusudan Gupta, 28 October 1836. Gupta becomes the first Indian to dissect a core. He was assisted by Rajkrishna De, Umacharan Sheth, and Darakonath Gupta, and Navin Chandra Mitra, students of all other students of the medical college. And still his birthday was recorded in West Bengal as the day of the anatomy. Now the next question, very important. This is asked sometimes in the oral examination. The term anatomy comes from the Greek word meaning to cut open. Tomy means cut and apart. And when you dissect, you cut and you slide the skin and other structures apart so that you can view. So from that region, from that meaning, the word anatomy cut. So ana means apart, tomos means to cut. So anything to cut and apart so that you view the internal structure, which is called anatomy. So word coming from the Greek word meaning to cut open or cut apart. So it is coming from ana and tomos. So ana plus tomos means anatomy. What is the origin? Greek. Just 2 lines you remember, write down. But not more than. Don't go headache to reading so many things about the dissection. Now, what are the subdivision? Again, not very asked in the examination, but you have to know. What are the subdivision of our anatomy? That is a gross anatomy. Usually we read, we dissect the body, we taking the bone classes. It is the gross anatomy. Grossly, we know the subject anatomy. So we call it gross anatomy. It is the one aspect of anatomy. There are so many other aspects of anatomy. So anatomy is not a simple thing. To know the body and something, anatomy extension is now beyond far. Next is your histology classes. Microscopic anatomy. We take the tissue from the body, usually from the living body. You process it so that it may be kept without any destruction of the tissues, the cells. Then you stain it and look under the microscope so that you can detail the microscopic structure, the cells and the boundary of the cell, different type of tissues so that you could identify the organ, normal thing. When you know the normal, then you can identify anything abnormal is present in this your microscopical anatomy of that particular organ or not. So then it is a purview of pathology. So anatomy is the normal microscopical anatomy. Pathology is the abnormal microscopic anatomy of the tissues affected by any disease. But without knowing the normal, how do you can know the abnormal? So that very vital. First, if suppose you take a tissue by some nursing personnel or some other, and they take the formalin solution and put the tissue on that particular vial, right? And by mistake, he or she written instead of liver, he written spleen. And you know it is a splenic tissue and you see, but you don't know normal. How do you know? And you think it is a spleen, you will give then a wrong report. That it is an abnormal. Liver is not like that. But actually it was a spleen. Or other way around, actually it was a liver. Sorry. Actually it was a liver. But wrongly, the sister or the technician written it the spleen. You think that it is the spleen, so no, it is not the liver. So you too written the grossly abnormal report. Grossly abnormal, make the normal person a disease. Otherwise, this is a normal person. So that you don't, even in X-ray, you don't not only depend the left L or R, you will be seen in your X-ray, in chest X-ray. L means left side and the R means right side of the X-ray plate. But if you depend on particular of the L and R, sometimes you will be put in a great trouble, because sometimes wrongly instead of L they put the R. So you have to know the side. Don't depend upon the L and R, that. So that similarly, you have to know the normal anatomy, microscopical anatomy. Then and then you can know the abnormal. So it is the, when you see the microscopical anatomy of a tissue or an organ, we call it microscopical anatomy. Other name of the microscopical anatomy is the histology, because histos means tissue, logis means knowledge. So histology. So gross anatomy, microscopical anatomy or histology, and developmental anatomy, very important for your examination purpose. There is the embryology. How from the zygote we develop. Not only this developmental anatomy stops after the birth of the baby, it may extend further post birth also. Complete development of an tissue, of an person includes the developmental anatomy. So other name of this developmental anatomy is the embryology. We normally call it embryology. It is in your subject. So gross anatomy, histology, embryology. You have to read. Last, specialized branches of anatomy. There are so many specialized branches are there, that will be other way you will write. There are so many nowadays is developed. I am not going into detail. It will take your time. So you have to know the thing. At the last, it is not written. That is another important for your examination, and possibly 15 marks. 4 into 5 are allotted to a subject known as the explain why. Mainly depends upon an applied anatomy or clinical anatomy. Nowadays it is important. Explain why thyroid gland, why moves with deglutition. Suppose there is a swelling in the thyroid gland, which is situated here. You look, there are swelling is there in the midline of your neck. It may be thyroid, it may be lymph node, it may be dermoid cyst. Then how do you differentiate in a bedside? You ask the patient to deglutate, mane dhok gelo, or put a glass of water and ask him or her to take this water. And if you see while he is swallowing, the swelling is moved up and down with deglutition, you must, I cannot say, in medical science there is no word like must. There is an every chance of having, it is not a lymph node, but a thyroid swelling, because anatomical explanation is there that thyroid swelling moves up and down with swelling or deglutition. So in this type of questions are come. Why inguinal hernia is common in female? There are anatomical explanations are there. You will learn. So this is known as the clinical anatomy. So this is another part of type of anatomy, special anatomy. There are other special anatomy, radiological anatomy, X-ray, CT scan, ultrasound. These are known as the radiological anatomy, or this is on the special branch of anatomy. There are so many branches of anatomy, of which what I am tell, it is included within your course, and you have to appear this subject in the examination. All this what I have told. There are other way we divide the anatomy, that is the regional anatomy and systemic anatomy. What is regional anatomy? Anatomy by the regions. Means, suppose this is, we call it the superior extremity. So everything from the skin, artery, vein, nerves, bones, only in the superior extremity are dealt in a subject. So it is the regional anatomy of the superior extremity. Similarly, thorax. The thorax, the skin of the thorax only. The underlying pleura, underlying muscles, underlying lungs, underlying heart, descending thoracic aorta, everything. Only the sub organs limited to the thorax only. We deal, when it is deal only the thoracic region, so it is known as the regional anatomy of the thorax. So this is the regional anatomy, right? Last, another is the systemic anatomy. Systemic anatomy means gastrointestinal system. Suppose there is a cell in the unit of an house, we call it the brick. Similarly, unit of our body is the cell, right? Unit of our body is the cell. Cell, multiple cell doing a similar function, we call it tissues. And tissue ultimately form organs. For example, in liver, liver consists of number of cells, different types of cells. And what is the difference between cell and tissues? Cells means a accumulation of cells when perform a particular function, we call it tissues. For example, epithelial tissue. There are epithelial cells are there, and this function is to only to cover. There are some other functions also, secretion, etc., but the main function is the cover. Similarly, connective tissue consists of number of cells, but their main function is to connect and giving the structural support to other things. We call it connective tissue. Muscle tissue. The muscle cells constitute the muscle. So there are 4 basic types of tissues are there. আজকে থেকেই জেনে নাও। সেটা হচ্ছে epithelial tissue, connective tissue জানো তোমরা 12 এ পড়েছো, muscular tissue and neural tissue or nervous tissue. So these are the tissues. Tissues form organs. Organs ultimately form system. So for example, gastrointestinal system. So it starts from mouth, then pharynx, then esophagus, then stomach, intestine, ultimately anus out. So when we read in this fashion, not localized by the regions, we call it systemic anatomy. Everything we read about, not dependent, we don't depend upon the regions, only esophagus, read only, because, for example, esophagus not only present in the abdomen, but also present in the thorax. So we have to read all things. So it is not out encircled by an regional anatomy. So we call it systemic anatomy. Another is it is included in your practical, that is a surface anatomy. You have to draw some surface projection of an artery. You have to draw, and there is an artery is there known as the ulnar artery. By means of a French chalk, you have to draw the ulnar artery there. There is a point. You take a French chalk point, and the examination like point the coracoid process. So you have to put a mark here. So fill it and put. So this is known as the surface anatomy. Very important of the surgeon when before going to operate. He knows the surface anatomy, then he can proceed. Even in your operation table, sometimes they draw the surface anatomy first, then he proceed. So this is the surface anatomy. There is a cytology, study of the cell. Histology, I have already told you. And embryology, I have told you. Pathological anatomy, molecular biology, already you know. I am not going into details. Now, that's the most important. This is starts of the vital chapter, not for writing anything. To know, it is the knowledge. You have to acquire things, not important in examination, but without knowing you cannot pass the examination. Some anatomical terminology is there. Sometimes it may be difficult to understand, but when time goes, you will be master of everything, right? Within few days, you will be master of this terminology. So human body, first of all, know, only make it very clear. What is the anatomical position of a human? Make it any position like that. He erect, head erect, standing in a straight posture. Posture means the position with eye looking directly forwards. both eyes are parallel and looking directly forward, right? Standing, head erect, eye looking forward. আমি তোমাকে দেখছি না কিন্তু আমি ওখানে দেখছি। And most important, palm looks like that. We call it supinated. And this is the palm of the hand. It is called the dorsum of the hand. ভালো করে দেখো, this is the palm of the hand and this is the dorsum of the hand. And palm of the hand is looking forward. Forward মানে সামনে, anterior. So lamb looking forward, eyes and feet should be close together. And position is that. It is known as the anatomical position. And hands will be hanged by the side. Elbow known as the extension. What is extension, flexion? I will come just now. So this is the anatomical position. Now, in anatomy, very important is the relations of this. Anything, relation means ki, suppose this is on the table. Hey, this is on the table. This is on the table. This is on the table. And I am standing on the right side of the table. My hand is, two hands is there. Suppose, bhalo kore dekho. Two hands. One is here. In relation, suppose it is the body. Human body. My left hand is there. Another hand is there. So we call it this hand. And this is the mid. This is the midline. Midline of the body. This is the, this pen is situated in the midline of this table. ভালো করে এটা লক্ষ্য রাখো, very important. So I put my left hand here. So my left hand is in the midline. And my right hand is here. So I cannot call it, my right hand is right side of the my left hand. বলতে পারি তো? আরেকটা ভাবে বলা যায়, my right hand is lateral to my left hand. Lateral, এই টার্মসটা এখন থেকে জেনে রাখো, lateral মানে মিড লাইনের সাইডে যেটা থাকবে তাকে বলি আমরা ল্যাটারাল। আরেকটা ওয়ার্ড ইজ ইম্পর্টেন্ট, ইট ইজ দ্য মিড লাইন। মাই হ্যান্ড ইজ দেয়ার অ্যান্ড লেফট হ্যান্ড ইজ দেয়ার, তাহলে এইটা কোনটা ল্যাটারাল? রাইট হ্যান্ড ইজ ল্যাটারাল অ্যান্ড নাও এটা কি বলবো, এটা তো মিড লাইনে নেই, মিডিয়াল। সো উই কল ইট মিডিয়াল। আরেকটা টার্ম জেনে রাখো, সেটা হচ্ছে ভেরি ইম্পর্টেন্ট। এরপরে ধরো superior and inferior. Superior means above and inferior means below, right? Superior means above, inferior means below. তাহলে এবারে আচ্ছা, এটা গেল সুপিরিয়র। তাহলে আমার সুপিরিয়র কোন দিকটা? হেড অ্যান্ড লেগ বিলো। এবার আমি শুয়ে গেছি, তো দুটো তো একই লেভেলে, কোনটা সুপিরিয়র কোনটা ইনফিরিয়র? তাহলে আই এম কমপ্লিটলি লাইং ডাউন, হুইচ ওয়ান ইজ সুপিরিয়র, হুইচ ওয়ান ইজ ইনফিরিয়র? কেন? একই লেভেল তো। আই এম লাইং ডাউন। ব্যাপারটা সিম্পল, ব্যাপারটা ইজ ভেরি সিম্পল। Whatever is the position of the body, we must think it in anatomical position. আমরা ওটাকে ভেবে নেব অ্যানাটমিক্যাল পজিশনে কোনটা সুপিরিয়র কোনটা ইনফিরিয়র ছিল। পজিশন যাই থাকুক, all relations are to be thought in terms of anatomical position. আমরা বডি শুয়ে আছে, I will imagine that body, if body is normal anatomical position, what is the superior, what is the inferior, আমরা ওটাকেই সুপিরিয়র বলবো। আমরা বডি শুয়ে আছে, হেড অ্যান্ড টেল বা ফিট দো ইন দ্য সেম লেভেল, উই হেড ইজ অলওয়েজ সুপিরিয়র, বিকজ ইন অ্যানাটমিক্যাল, এই জিনিসটা ক্লিয়ার হয়েছে? যেভাবেই থাকুক বডিকে আমরা কল্পনা করব কোনটা সুপিরিয়র। সাপোজ বডি ইজ লাইং, right? Body is lying down, our abdomen is above. তাহলে অ্যাবডোমেনটা কি সুপিরিয়র? নর্মালি কি আরেকটা টার্ম জেনে রাখো, anterior and posterior. এই এক্সাম্পলটা it will be very clear, make it clear. Make it very clear. Anterior means in front, in anatomical terms. Posterior or back means this. Posterior means backside, right? Ever, I am lying down. I am lying down. So my abdomen is superior, obviously. উপরে উঠে গেল তো, ব্যাকটা কি হলো? ইনফিরিয়র হয়ে গেল। কিন্তু যখন আমরা রিলেশন লিখব অ্যানাটমিতে, অ্যাবডোমেন ইজ অলওয়েজ অ্যান্টিরিয়র, অ্যান্টিরিয়র অ্যান্ড অ্যান্টিরিয়র। যদি লিখতেই হয় সুপিরিয়র, আমাদের লিখতে হবে তখন যে ইন লাইং ডাউন পশ্চার অ্যাবডোমেন, অ্যান্টিরিয়র অ্যাবডোমেন ইজ superior to posterior abdomen, সেরকম আমরা বলি না, আমরা সব সময় অ্যানাটমিক্যাল টার্মসেই বলি। সুতরাং অ্যাবডোমেন অ্যান্টিরিয়র মিনস অলওয়েজ অ্যান্টিরিয়র। সো আমাদের এখানে একটা রিজিয়ন রয়েছে, ভালো করে এটা বোঝো, নইলে কিন্তু পরে অসুবিধা হবে বুঝতে। এইটাকে বলে ইঙ্গুইনাল রিজিয়ন। এটাকে যখন ডিসেকশন করব আমরা স্কিনটাকে, when you cut the skin, taking it apart and we find a canal inside, we call it inguinal canal. So inguinal canal has got boundaries, anterior boundary, posterior boundary, roof and floor. It is a canal like a, so it is, suppose this is, so this is got an anterior boundary, suppose this is the canal, right, through which spermatic cord comes down. This is the canal and we have to know the boundaries of the canal. So in anatomical term, this is the, look my body, this is the anterior, this is the posterior, this is the superior, this is the inferior. Now patient is lying down. So whether this boundary will be superior? No, it is always anterior. Because in anatomical position it is anterior. Clear হয়েছে এটা? ভালো করে এই গন্ডগোলটা ভাবতে পারে, যেটা আমার হয়েছিল। anatomical position এ this region is anterior. এবার আমি শুয়ে গেলাম, this region gets superior. So whether the boundary of the inguinal canal will be changed? No. Anterior means always anterior in anatomical position. Clear হয়েছে এটা? Clear? তাহলে এবার একটুখানি রিক্যাপিচুলেট করি, ছবি দেখে তাহলে আরো ক্লিয়ার হয়ে যাবে। So some anatomical directions: superior, inferior, anterior, posterior, dorsal, ventral, proximal, distal, medial and lateral. এইগুলো হচ্ছে অ্যানাটমিক্যাল টার্মস। তোমাকে আজকে হয়তো আগের ক্লাস থেকেই শুরু হয়ে গেছে। অ্যানাটমিক্যাল পজিশন আমি বলে দিয়েছি, স্ট্যান্ডিং আপরাইট, ফেসিং দ্য অবজারভার, হেড লেভেল, আইস ফেসিং ফরওয়ার্ড, ফিট ফ্ল্যাট অন দ্য ফ্লোর, আর্মস অ্যাট দ্য সাইড and palms and turn forward. বলে দিয়েছে, করব না। Anterior refers to a structure being more in front than another, and always these terms anterior and posterior means comparison. This bottle is anterior to the pen. Come on. So this bottle is anterior to the pen and this pen is posterior to the bottle, right? This pen is posterior to the bottle, but this pen is anterior to the duster. And this pen and duster is posterior to the bottle. So it is always a comparison. Eta posterior মানে it is not always posterior. So এইটা clear হবে। কেমন, রাইট? এটা গেল। তার জন্য লিখতে refers to a structure being more in front than any another structure of the body. যেটা বেশি সামনে সেটাই anterior, যেটা বেশি পেছনে that's posterior. Always a comparison. Medial and lateral. This is the midline. So it is the midline position. Anything, suppose দুটো হলো, so this is the midline, so it is lateral to the midline. আবার যে আরেকটা হয়ে গেল আবার this pain is lateral to the this is the remote, but remote is medial to the pain. Remote is medial to the pain, কারণ এটা more midline এ যেটা থাকবে সেটাকে আমরা medial বলবো. Which is more medial, which is more towards midline, we call it medial. Always comparison, and comparison is not fixed one. It is not static, depending upon the structures, it varies. তাহলে medial lateral clear, right? Distal and proximal. Again comparison. Distal means এই যে আমাদের বডি, কেমন বডির কাছে ক্লোজ যেটা রয়েছে দ্যাট ইজ প্রক্সিমাল, আর যেটা দূরে রয়েছে দ্যাট ইজ ডিস্টাল, বা সেন্ট্রাল পয়েন্ট অফ দ্য বডি সেখানে যেটা ক্লোজ রয়েছে ইট ইজ প্রক্সিমাল, দ্যাট ইজ ডিস্টাল. ফর এক্সাম্পল, দিস ইজ দ্য হ্যান্ড, দিস ইজ দ্য হিউমেরাস, this is the radius bone, this is the ulna bones. Come on, left side. Okay. So tell me, humerus is proximal or this is the ulna? Ulna is proximal, humerus is proximal, right? So ulna is distal to the humerus. Abar this is known as the metacarpal bones. So ulna is proximal to the metacarpal. Ulna is proximal to the metacarpal. It is the metacarpal bone, and these small bones we call it phalanges. So ulna is proximal to the ulna is proximal, very good, metacarpal. তাহলে proximal distal clear. এই হচ্ছে আমাদের এরকম ছিল. এইবারে বলো which one, this is the radius, this is the ulna, which one is medial, which one is lateral? Ulna medial and radius is lateral, right? তাহলে ক্লিয়ার হচ্ছে আস্তে আস্তে, খুব তাড়াতাড়ি ক্লিয়ার হচ্ছে. ইউ আর ভেরি ইন্টেলিজেন্ট, আই ডু থিঙ্ক. আগের ব্যাচে আমার একটু কষ্ট হয়েছে. এরপরে আরেকটি ইম্পর্টেন্ট, এগেইন কম্পারিজন, সুপারফিশিয়াল অ্যান্ড ডিপ. সাপোজ উই কাট দিস হ্যান্ড, দ্য স্কিন, then we find a woolly structure, we call it superficial fascia. Then we cut the superficial structure, we find a dense membrane-like fascia, we call it deep fascia. So we call it skin is superficial to the superficial fascia, and superficial fascia is deep to the skin. But superficial fascia is superficial to the deep fascia, and the deep fascia is deep to the superficial fascia. গন্ডগোল হচ্ছে. এই ছবিটা দেখো, this is the skin, ভালো করে লক্ষ্য করো, this is the skin, হ্যাঁ এটা স্কিন, রাইট? এটাকে বলা হয় সুপারফিশিয়াল ফাসা, সাবকিউটেনিয়াস টিস্যু. স্কিন কাটলে এটা দেখতে পাচ্ছি, সাবকিউটেনিয়াস টিস্যু. তারপরে একটা রয়েছে ডিপ ফাসা, তাকে বলে টেম্পোরোপ্যারাইটাল ফাসা, এখন জানার দরকার নেই. ডিপ ফাসা, তারপরে একটা এরিওলার টিস্যু রয়েছে, তারপর মাসেলস রয়েছে. এইবার শোনো, স্কিন ইজ, এটা হচ্ছে সাবকিউটেনিয়াস টিস্যু, এটা স্কিন ইজ সুপারফিশিয়াল টু দ্য সাবকিউটেনিয়াস টিস্যু. স্কিন ইজ superficial to the subcutaneous tissue, and subcutaneous tissue is deep to the skin. Subcutaneous tissue is deep to the skin. মনে আছে? এরপরে এই muscle is deep to the subcutaneous tissue, right? And subcutaneous tissue is superficial to the muscle, and muscle is both muscle and superficial subcutaneous tissue is both deep to the skin. তাহলে এবার একটা বলি, এই জায়গাটা ইচ্ছে করে অমিট করলাম. এটাকে বলা হচ্ছে loose areolar tissue. So loose areolar tissue is deep to the muscle and muscle or not? Loose irregular tissue, whether it is a deep to the muscle? No, it is superficial to the muscle. Our muscle is whether deep loose irregular tissue is superficial to the skin? No, it is deep to the skin, but it is superficial to the muscle. Comparison. মনে থাকবে? আচ্ছা, এটা হয়ে যাবে আস্তে আস্তে। এই আইডিয়াগুলি ইম্পর্টেন্ট। এরপরে হচ্ছে পজিশন, এটাকে ভেন্ট্রাল এবং এই দিকটাকে বলা হয় ভেন্ট্রাল, এদিকে ডরসাল। Especially it is more important in fetus when it is remain floated in the amniotic fluid, because fetus is always floating. It is, there is a no anterior posterior something, always floating, কখনো এরকম কখনো ওরকম মায়ের ঘুরছে, so that we don't usually, don't বলবো না, sometimes we usually do not use the term anterior and posterior, superior and inferior. In fetus we usually term, instead of anterior we call it ventral, instead of posterior we call it dorsal, instead of superior we call it cephalic, cephalic means towards the head, and instead of a কি বললাম inferior we call it caudal, মানে লেজের দিকে, তলের দিকে। ছেড়ে দাও, গুলি হয়ে যাবে, চিন্তা নেই, যেটা ইম্পর্টেন্ট। আচ্ছা, this is the position. When we lying flat with abdomen up, we call it supine position. When we head down, we call it prone position. And one side of the body, we call it unilateral. And when you taken the both side, we call it bilateral. Suppose, and another name of uni is a hemi, half. In Latin it is called the hemos, so it is hemi. Suppose there is an one hand is paralyzed, we call it hemi paralysis by hemiplegia. Another name of paralysis is plegia, hemiplegia, অথবা বলতে পারি unilateral plegia, one side যদি যায়, whole body of the one side, we call it hemiplegia. So, and when both hands are চলে গেল, both legs চলে গেল, we call it bilateral. দুটো সাইড চলে গেল, bilateral. আরেকটা কম্পারিজন ওয়ার্ডটা এখনই জেনে রাখো, ভেরি ইম্পর্টেন্ট, ইপসিল্যাটারাল অ্যান্ড কন্ট্রাল্যাটারাল। ইপসি, IPSI, ipsilateral. Ipsilateral means same side. Sometimes it is happened in certain region of the nervous tissues. There is an sensory loss. What is sensory? What is motor? Sensory means there are some, we receive some sensation from the environment. An environment in our world, there are two environments we are always encountering. We are encountering two environments. One is the external environment and one are inside internal environment. There is an internal environment is also there. So we are encountering always two environment, external environment and internal environment. Suppose sometime one give prick, so it is the coming from the internal external environment. And sometimes stomach is stressed and there is a pain, stomach pain, we call it encountered from the internal environment, whatever it may be. Usually there are so many sensory stimulations are there. Pain, touch, temperature, this is the most vital sensory stimulation. Pain, touch, and hot and cold. Besides, there are some other sensory also known as the proprioception. Proprioception means suppose we close an eye, we have got idea, even closing our eye, we can idea that I am standing, I am, my head is such, my leg is erect, not flexed, but not bended. So this idea and is known as the proprioception. মানে স্পেসের মধ্যে যেটা রয়েছে সেই আইডিয়া, 2 পয়েন্ট ডিসক্রিমিনেশন, দুটো পয়েন্ট যদি আমার চোখ বন্ধ, কেউ টক করে দুটো হাত লাগাচ্ছে, আমি বলতে পারছি দুটো পয়েন্ট লেগেছে। এই যে আইডিয়াগুলি আসছে, পরে শিখবে। জয়েন্ট সেন্সেশন, আমি বন্ধ করেও কোনো সেন্স না হলেও বুঝতে পারছি আমার এই পাটা বেন্ড হয়ে আছে, এই পাটা স্ট্রেট। এইগুলিকে আমরা বলি প্রপ্রায়োসেপশন। দেয়ার আর সো মেনি সেপশন রয়েছে। কেমন, আমি চোখ বন্ধ করে বুঝতে পারছি আমার মাথাটা সোজা রয়েছে, আমি এবার বুঝতে পারছি মাথাটা বেঁকে গেছে, বা আমি উল্টো হয়ে রয়েছি, আমি ঝুলে গেছি, সার্কাসে যেরকম হয় ওরা বুঝতে পারে যে আমার হেড ডাউন, সেইগুলি হচ্ছে প্রপ্রায়োসেপশন আইডিয়া রয়েছে। স্পেসে মধ্যে যে আমরা রয়েছি, দ্যাট ইজ নোন অ্যাজ আ প্রপ্রায়োসেপশন। সো দেয়ার আর sensoriception. Suppose there is a loss of pain touch temperature of this superior extremity. This is called the superior extremity. This is called the inferior extremity. আমরা ওই হ্যান্ড ফ্যান্ড বলি না, আমরা বলি superior extremity and inferior extremity. Suppose there a person come to you and sometime it will come, there is a loss of sensation of the superior extremity and paralysis of the left side. মাসল প্যারালিসিস হয়ে গেছে, আমরা তখন বলবো এটাকে কন্ট্রালেটারাল এফেক্ট। কন্ট্রালেটারাল, রাইট সাইডের একরকম, লেফট সাইডের একরকম, সেটাকে বলবো আমরা কন্ট্রাল্যাটারাল। আর যদি সেম সাইডে গেল, এইদিকেও গেল এদিকেও গেল, আমরা বলবো ইপসিল্যাটারাল। অনেক সময় এখানে ক্রেনিয়াল নার্ভ যে রয়েছে, ফেসের এই দিকের প্যারালিসিস হয়েছে আর হাতের উল্টো দিকে প্যারালিসিস হয়েছে, দেন উই কল ইট কন্ট্রাল্যাটারাল প্যারালিসিস। আর যদি সেম সাইডে হয়, ইপসিল্যাটারাল প্যারালিসিস। এই টার্মস গুলি ভেরি ইম্পর্টেন্ট। আচ্ছা, সুপাইন, এটা সুপাইন পজিশন, বুঝতে পেরেছো? দিস ইজ দ্য প্রোন পজিশন, দিস ইজ দ্য রাইট ল্যাটারাল পজিশন। বেডের কাত হয়ে রয়েছে, বেডের কাছে রাইট সাইডটা টাচ করেছে, বেডের রাইট সাইডটা টাচ করেছে, we call it right lateral. যদি left side touch করে, left lateral position. This is the most important position in obstetrics and gynecology, and also in surgeons in some operation near the anal. This is the lithotomy position. Lithotomy position, patient lying on the bed with, and this is known as the gluteal region or buttock, on the edge of the lower edge of the table. Hip is 90-degree flex. Flex means bending. Hip is কতটা flex হবে? 90 degree. Knee joint is 50 to 70-degree flex. Come on. And the leg is rest on a leg rest. সেইটাকে আমরা বলি, and thigh is abducted. Abducted মানে কি? এই পাশটা সরে যাওয়াকে আসবো আমি, এটাকে বলি abducted. সেইটাকে আমরা বলি লিথোটমি পজিশন। এই পজিশনটার নাম হচ্ছে লিথোটমি পজিশন। লিথোটমি পজিশন, ইট ইজ অ্যাবডাক্টেড 30 ডিগ্রি। দিস ইজ নোন অ্যাজ দ্য নি চেস্ট পজিশন। নি চেস্ট পজিশন, নি আর উল্টো প্রোন টাইপের কিন্তু উঠে রয়েছে, নি আর চেস্ট ক্লোজ টুগেদার, নিটা একদম চেস্টের কাছে লেগে রয়েছে, knee chest position. This is known as the Trendelenburg position. Head and low, lower and high. This is known as the Trendelenburg position. এই Trendelenburg অনেক টার্মে রয়েছে, ইউনিল্যাটারাল, বাইল্যাটারাল, ইপসিল্যাটারাল, কন্ট্রাল্যাটারাল, যেটা বলেছি তারই ছবি রয়েছে। ক্লিয়ার? এই তো লাস্ট, আজকে আর টাইম নেই। অ্যানাটমিক্যাল প্লেনস ভেরি ইম্পর্টেন্ট, বারবার আসবে। coronal, sagittal and cross sectional or transverse plane. এই যে হেড, এই যে সুচার, সুচার মানে কি? জয়েন্ট। এখানে একটা বোন রয়েছে, এটাকে বলে ফ্রন্টাল বোন, আর দুপাশে দুটো রয়েছে প্যারাইটাল বোন। এই ফ্রন্টাল এন্ড প্যারাইটাল বোনে যোগ রয়েছে, একটা সেলাই করার মতো রয়েছে, তাকে বলে সুচার। এই সুচারটার নাম হচ্ছে স্যাজিটাল সুচার। কি সরি কোরোনাল সুচার, আই এম রিয়েলি সরি, কোরোনাল সুচার। কি বলে? কোরোনাল সুচার, CORONAL। আর দুটো প্যারাইটাল বোন is connected by a suture, we call it sagittal suture. So any plane এই sagittal suture দিয়ে পাস যদি করে প্লেন, বা আমি যদি করার দিয়ে কাটতে থাকি লোকটাকে, এরম ভাবে কেটে we divide it into any portion right and left দুটো আলাদা ইকুয়াল রাইট অ্যান্ড লেফট ভাবে ভাগ করে দিলাম লোকটাকে কেটে এইদিকে স্যাজিটাল সুচার দিয়ে এরম ভাবে তাকে বলবো যে প্লেনটা দিয়ে কাটা হলো which plane which we cut we call mid sagittal plane mid sagittal এই টার্মটা ইউজ করতে হবে mid sagittal plane মিড লাইন দিয়ে কাটছি mid sagittal plane আর এনি প্লেন sagittal সুচারের প্যারালাল দিয়ে কাটছি not at the midline it is known as the parasagittal or simply sagittal plane যাতে রাইট অ্যান্ড লেফটে ভাগ হবে বাট রাইট পার্টটা হয়তো ছোট লেফট পার্টটা বড় হবে বা লেফট পার্ট ছোট হবে রাইট পার্ট বড় হবে সেইটাকে বলি আমরা প্যারাসেজিটাল অর সিম্পলি স্যাজিটাল প্লেন আর যদি মিড লাইনে যায় তাকে বলি মিড স্যাজিটাল প্লেন আর এইখান দিয়ে যদি আমি কাটি যাতে অ্যান্টেরিয়র অ্যান্ড পস্টেরিয়র পজিশনে ভাগ হয়ে গেল একটা মানুষ তাকে বলি করোনাল প্লেন এই প্লেনটাকে বলা হয় করোনাল প্লেন এবং এইটা হচ্ছে স্যাজিটাল প্লেন দেন ইউ ক্যান ইজিলি আন্ডারস্ট্যান্ড দ্যাট করোনাল অ্যান্ড স্যাজিটাল ইউজুয়ালি কাট অ্যাট 90 ডিগ্রি দুজনের মধ্যে রিলেশন হচ্ছে 90 ডিগ্রি আর এনি পজিশন যে ট্রান্সভার্সলি কাটি দেন উই কল ইট হরাইজন্টাল প্লেন সো মেইনলি আমরা আরো পজিশন আছে অবলিক প্লেন দরকার নেই দিস আইডিয়া করতে হবে ভেরি ভেরি ইম্পর্টেন্ট পরে আসবো কেন ইম্পর্টেন্ট especially today is the today of the CT MRI so whether if you have got an idea about this plane you cannot understand go to the MD radiology or something because in one section suppose we take a ray here we find a liver only one part of the liver then take another view there another view there then another view there another view in this slide rotate the in this way then computer associated is all the structure and give the whole body whole structure of the ki bole of this organ so that you have to idea about the this plane so very important sagittal mid sagittal coronal and transverse plane because যদি আইডিয়া না হয় তাহলে তুমি বুঝতেই পারবে না কোনটা কি কারণ এইটা it is looks in transverse plane one type and in coronal plane another type so you have to have idea about this plane this is the coronal plane same body coronal plane in the femur but suppose this is taken a transverse plane here femur in the coronal plane more or less whole of the femur you can see but in transverse plane you see only a rounded structure যেমন এই যে হিউমারাস যদি করোনাল প্লেন হয় কি দেখতে পাচ্ছি পুরোটা দেখতে পাচ্ছি কিন্তু যদি ট্রান্সভার্স প্লেন হয় অনলি রাউন্ড আইডিয়া করতে হবে ওটা থেকে দিস ইজ দ্য হিউমারাস আর যদি স্যাজিটাল প্লেন হয় প্যারাস্যাজিটাল প্লেন এরকম ভাবে কাটা যাবে দুটো মাঝখানে ম্যারোটা থাকবে সেটার অন্যরকম আসবে সুতরাং দিস আইডিয়া ইজ ভেরি ইম্পর্টেন্ট This is the sagittal plane of the CT of a brain. সেটা আবার পাল্টে যাবে coronal plane. Movements আজকে থাক 11টা বাজে so thank you very much কালকে এটা দিয়ে আমরা সুপেক্সে ঢুকে যাব কারণ টাইম ইজ ভেরি শর্ট